Assalamualaikum warahmatullahi wabarakatuh. My name is Fatina Jha binti Muhammad Amir Darus with matric number 1171510. I am one of the students under supervision of Dr. Muhammad Nazmi bin Abdul Manap. Today, I will enlighten all of you about my proposal on the topic a single nucleotide polymorphism in microcalpain at exon 14 associated with tenderness and marbling score in bovine meat. In this video letter will be further explained on introduction, literature review, materials and method, expected result, Gantt chart, and also references. As for introduction, beef is one of the food that we consume by society all around the world and well known as high protein content food. The quality of meat is difficult to define as it might vary based on the understanding of consumer and can be defined in different ways. Meat quality also can be determined by the amount of usable lean and palatability factors such as marbling. Marbling has significant link with beef juiciness, tenderness, and palatability. The use of single nucleotide polymorphisms, or we call as SNPs, as genetic marker, is one of the methods to improve the quality of meat. Many efforts were done by meat producers as well as researchers in order to improve meat quality. Several researchers have showed the association of microcalpain with marbling in bovine meat. This proposal has been made because of this problem. Nowadays, many consumers focus on meat quality when purchasing meat. Greater marbling score in beef give higher perception on palatability. However, the intramuscular fat can only be seen after slaughtering and cutting process, so there must be a genetic marker like SNP that can be used to predict variation of intramuscular fat in beef before slaughtering. The purpose of this study is to determine the involvement of microcalpain gene with the variation of marbling score in beef in order to determine its ability to produce desirable trait as gene marker. Hence, the objective for this study is first to determine the association of microcalpain gene with marbling properties in beef, second to analyze and compare the marbling score among the individual cattle in both Taurus species using suckling method, and third to amplify the single nucleotide polymorphism in microcalpain gene at exon 14 by using polymerase chain reaction. Next, for literature review, I will explain overall about this topic based on findings from previous researches on meat quality, calpain gene, and association with the SNP. Food and Agriculture 2014 has reported that quality of meat is generally interpreted by lean to fat ratio, and the visual identification of quality meat is based on color, marbling, and water capacity. One of the quality that were considered before buying the meat is the marbling score in meat. Based on Miller RK 2002, the IMF content has an impact on consumers' palatability because of the association of the marbling score and the juiciness of the meat. Next, Dan S. et al. 2013 state that the quality score is given for each carcasses based on the degree of marbling in meat. For the calpain gene, calpains are classified as cysteine protease which rely for their activity on calcium, and the mechanism of calpain play an important role in proteolysis and meat tenderization postmortem. Among all of the family in the calpain gene, two ubiquitous isoforms which are microcalpain and m-calpain become the main point because of the specific pattern showed in both genes related to muscle growth. For the association of SNPs in microcalpain, summarizing from all of the successful findings from the previous researchers, this gene might be used as a possible candidate gene in determining the marbling of beef. In the materials and methods, the first step is very important, which is the sample preparation, as this might affect it on the results if not handled accordingly. To keep the variable constant, 10 samples of beef will be selected within the same species, which is posteriors of the average of 24 months, with only the tenderloin part will be chosen. The samples later will be collected and stored at the temperature of -20 degrees Celsius upon reach at the laboratory. The method will be divided into three big groups, consists of analysis of meat quality, analysis for molecular DNA, and lastly the statistical analysis. For meat quality analysis, the chocolate extraction method and texture profile analysis will be done to determine the textural characteristic and fat content in meat. For molecular analysis, the experiment will be done is DNA extraction, DNA quantification, DNA amplification, electrophoresis including staining of ethidium bromide, and visualization of agarose gel followed by DNA sequencing. Lastly, further statistical analysis will be done to determine the association between SNPs in microcalpain with meat quality. The result that were expected in the study is first able to analyze the texture and intramuscular fat content in beef. Next is able to detect the presence of SNPs in microcalpain of bovine meat. And lastly, able to detect the association of SNPs at exon 14 in microcalpain gene with the intramuscular fat content of beef. For this segment, this is the Gantt chart which show the total up of work and time that I'm planning to spend in this research and thesis writing. Last but not least, this is some references on the previous findings and research that were used to complete this proposal. And thus conclude my presentation. Thank you and Assalamualaikum.